right, we'll get started. Are you guys okay of doing mTOR right after the fat loss? So we can do muscle building right after losing fat. Today, we'll just do the metabolism beginning to end. You have fat somewhere in your stomach, in your hips, in your triceps, in your whatever, in your chin, wherever your fat is. And what happens to that fat from beginning to end of consumption, of mobilization and then consumption. So we'll do beginning to end with that today. And we'll do just nothing but tips and strategies, whether it's exercise, sleep, supplements, diet, other behaviors that have support in the literature for fat loss, stuff that's prescriptive, stuff you can actually do. But today is just metabolism beginning to end. Points are updated. Be thinking about your team names. Let's say by Friday. We have team names by Friday. Winner gets a third of a point or something. Case studies, just make sure you can do them. I put the case studies up on the clinicalphys.com. If you just scroll down, or no, I put it up on Canvas. And when we're getting close to the exam, I will put a PDF with all of the case studies and all of the review slides and all of that at that Clinical Phys page. Be able to do the kid one, just all the stuff we've been covering. Make sure you can do both of these, right? Where you can stratify into those 6 categories, as well as within the 6 categories, figure out the number of risk factors, blood pressure, and vasodilation, cholesterol, the lipid profile, the interaction with the immune system, how plaque gets deposited, atherosclerosis, how that gets deposited, that stuff, and what foam cells are, what the crack in the plaque, what that leads to, all of that, the effects of exercise. Not everybody is a good responder to exercise. BMI, I'm just going to start clicking. We're good with all of this stuff. And we're going to be talking more and more about this, especially considering we'll be doing the anabolic part of growth right after we do the catabolic section. So on Friday, we'll start getting into anabolism, muscle growth, hypertrophy, instead of fat loss. But right now, that insulin receptor and that signaling, we talk about bringing sugar into the cell, but insulin does a lot of other things too. It's going to be horrible for fat loss. Really the biggest barrier to losing fat that we have inside of us. Outside, you could say things like being super stressed or eating way too much, like way too many Fig Newtons or too much cake. Okay, but in terms of our own biology, the biggest barrier to fat loss is the insulin. But it's also very anabolic. And so when we get into muscle building, we'll talk about insulin a little bit more. Remember, insulin, you are reassembling glycogen, diabetes, metabolic syndrome, make sure you can do that stuff. The difference between insulin and glucagon, we'll keep talking about that in the muscle building section. But those beta cells, you have way more beta cells than you do alpha cells. Beta cells are the ones that release insulin. Glucagon comes out of the alpha cells. Remember, everything goes to the liver. It's going to go through the liver first before it gets to systemic circulation. Anything you eat, anything that comes out of your pancreas, the drugs, but you don't need to know them. Exercise, that's what you need to know. Other than metformin, how that works, because we're looking at one of those mechanisms of that drug is AMPK. AMPK is going to keep coming up in the anabolic section when we start talking about mTOR, when we're talking about hypertrophy, muscle growth, recovery from exercise. AMPK is a big impediment to hypertrophy, but it's great for diabetes. It's almost always great for cancer. It's basically always great for cardiovascular disease and longevity, all-cause mortality, stuff like that. AMPK is wonderful, especially for diabetes. So remember, we're halting glycogen synthesis. We're turning on through hexokinase and PFK glycolysis, so we're consuming the carbs that we get in, and then also things like more long-term stuff, mitochondrial biogenesis. Let's create some more mitochondria, and this is where it's going to antagonize hypertrophy is in protein degradation, which is all of this. A lot of cancer regulation there, but protein degradation we're going to get in the way of hypertrophy. You know the kid stuff. Just be safe with kids, design it right, and all of those things that people say are misconceptions. None of it's really true. You're going to improve any way you measure it. You know, it's jumping, it's throwing, it's technical skills, it's strength, it's whatever it is. Kids are going to improve with resistance training. And the typical profile in the literature of what we're seeing enhancing kids' performance is safe. Okay. So the body types, you don't really, this isn't something that's critically important, but we all have different bodies. And just the general shapes of those bodies differ. I mean, look around the room, look around campus, look in your family, look, I mean, just go wherever you go, just look at people, and their bodies are shaped differently. In general, the few shapes we talk about are the ectomorph, right? The minute bulls of the world, the mesomorph, the Usain Bolts of the world, and the endomorph, the Jack Blacks of the world. Those few body shape. But obviously, it's a spectrum, right? There are gradations, and there are infinity body types. Every single person has a different body type. And we see that inside too with our hormone profiles. Now, the way we're measuring these things, body composition, it's a helpful thing to know. DEXA, that's a very good way of assessing. You could do FIT3D, you could do the hydrostatic tank, you could do bioelectrical impedance, varying levels of accuracy in all of those. Okay, so what's the BMI?
Height and weight.
Yeah, it's just height and weight. It's a ratio of height and weight. And what are the numbers that it tells us?
Like 20 to 25 is normal, 25 to 29 is overweight, and 30 plus is obese.
Perfect. Yeah, so we have these categories. We have a specific number, and then we have the categories that say, you are normal weight, you are overweight, you are obese, and then we get into these stages, 1, 2, 3 categories of obesity and getting into morbidity, levels of rotundity that predispose us to some complication.
3.
What's the limitation of BMI? Yeah, there's no consideration of your actual body composition. Where are you storing? Is that just meat, right? Is it just a bunch of like bone and sinew and whatever food you just finished eating, or is it pure adiposity? Between the two, 0.027. Okay, so with BMI, it's a nice, easy way of assessing things, but it doesn't really tell you anything. We like to think it does. We like to think that it tells us about our body fat percent. We like to think that it tells us these, I don't know, health risk and what your composition is, and then the association with illness. But we have The Rock, and he's going to be healthy obese. Probably, I'm assuming he's healthy obese. He'll be in that obese category, but his body fat percent is very low. We have Princess Diana, sort of. We have her head on someone else, but this would be that skinny fat, right? So we're looking at some midriff and a little bit of extra adiposity, abdominal adiposity in what would be a low BMI. Get a little bit of tricep chunk and a little bit of abdominal adiposity, and we start to look at BMI being inappropriate for that person in terms of disease class.
Like clinical practices for like APT, single APT, single line APTs?
I have no idea, but I love the question.
Yeah.
For the sake of how much weight that can be, I remember I was a middle schooler and I was a wrestler. And I was wrestling a guy with no legs, and we were in the same weight class. Yeah. Like, man, this is like, I'm like wrestling Nathaniel. I'm like a middle schooler. I'm like a tiny middle schooler. I'm wrestling like Nathaniel, but just without the legs. And so one has no idea how to kind of negotiate that contract, that sort of physical engagement. but it matters. Yeah, so this stuff really matters, and it's a great question. If the research doesn't exist, we should maybe figure out a way of doing amputational BMI correction.
Yeah.
That would be an easy publication if one does not already exist. But remembering this right here, waist circumference. Remember in ACSM where they say everybody with a BMI of 30 or higher, males, females, who cares, 30 or higher, that counts. But waist circumference, you could have a BMI that's normal. You could have Tyrion Lannister 0.4 BMI. But if your waist circumference for a male is 102 and for 8 centimeters, and for a female is 88 centimeters, now that's a risk class. So just abdominal adiposity, just visceral fat storage matters. And we see that in the literature. The reason that exists is when you have a higher waist circumference at the same BMI, mortality, all-cause mortality, we're seeing this elevation in risk. And obesity is not just diabetes. There's tons of metabolic complications, tons of cardiovascular complications. There's sort of an urgency to fix this problem. It really predisposes us. All right, let's relive this conversation. It has grown a little bit since our last lecture. All I said was you have a slow metabolism, right? So this is like the asshole who's criticizing people and unaware of the harm in his words. But the problem with that is, what is metabolism? Okay, I can't do it fast. Yeah, the sum of all chemical reactions in your body. You add them all up, right? Add up all of those chemical reactions. You're 1, okay. Plus 0.01. So you add them all up. Out of all those chemical reactions, some of them are catabolic, some of them are anabolic. And the anabolic ones, we're gaining weight, we're putting on pounds, we are growing. Catabolic ones, we're shrinking, we're atrophying, we are shedding the pounds. You sum all of these things up and you get your total metabolism. Cals in, cals out, all there is to it. Obese comes from Latin's, obedere, I think it's pronounced. There's a couple of different iterations of this word, but it literally means eating to excess. It means overeating, depending on the version of the word. But the root word of obesity, people talk about overeating. And so we have come up with this explanation for obesity, which is that you ate too much. Obese, you ate too much. The root word says it all. But if you think about it, what's a lipoma? Do you guys know?
A lipoma? Isn't that like it's your skin, it's a skin disease, and you get, like, it's a fat molecule, and then it pops up and you have to get it surgically removed by a dermatologist?
That's very specific. That's like, you've encountered this before. No, I know you personally, but you've encountered it in some societal context. Yes, that's what it is. Like with dogs, just if you have an old dog, pet that dog down, and you're gonna find a lump. If your dog is like, double digits in age, in human years, dog years, and pat it down, and you're gonna find a lump. That's a lipoma. It's a fatty little tuft of tumor. Now, don't eat anything. Just fast, starve, clear to death. Lipoma is still gonna grow. Calories in, calories out. Doesn't make sense, right? If tissues can grow in the presence of no calories at all, doesn't quite make sense. Why do all dieters, every single dieter who has ever dieted, why do they plateau? If you think about calories in, calories out, as from the prescriptive perspective, go on a diet, keep your exercise identical, do not change anything, down to the calorie, increase it actually, increase it. Let's say before dieting, you were burning 500 calories a day through exercise, and now you're burning 750 a day through exercise. You're still gonna stop losing weight. You cut calories and increase exercise, and you will stop losing weight. You will plateau. Calories in, calories out. If that were true, that scenario would be impossible. Why do males and females store fat in different places? Look around you. Everybody has a different, males and females in particular, have different fat storage. We call it gynoid fat. We call it android fat, different presentations of fat. If it weren't hormones bossing around your fat, if it were just like, well, you eat and you grow or you shrink, hormones are making a lot of decisions for you. You know, why do age and medications slow our metabolisms? Why do we gain weight as we age? Watch people cross a certain age threshold, like, man, I just can't keep the weight off anymore. I'm exercising more than I ever did. You know, I used to just eat whatever I wanted. Listen to people say this. You've all heard this from people, and you're like, oh, you're just old. You know, those are the words that old mouths make. But it's true. It's not like 100% of old people forever have always lied to everyone young. It's true. You reach a certain point, and the hormones start to change, and you're exercising more than you ever did. And you're eating less than you ever did. And you're gaining fat. Calories in, calories out. It doesn't make sense. There is no math. There's no equation in which that makes any sense. Or medications. Some are like, well, you know, I went on Depakote or whatever, or some you know, medication. And you'll watch all this weight gain. Did they change their exercise? No, they were more mindful about it, and they increased the amount of exercise because they knew that medication associated with weight gain. They watched their diet, and what happened to the scale? It goes up, it goes up, it goes up. When someone hits menopause, all right, let's put age in there. Well, what happens? They gain weight, right? Calories in, calories out is the most ridiculous short sentence to talk about how the body stores fat and how the body discards fat. So calories in, we can control this. That's something you can control. You can measure it on a little scale. You can put, you know, a certain amount of food in your mouth, swallow it, whatever. You can know that somewhere close to precise number of calories you've eaten. Calories out, you are in no control of that. You're in no control of calories out. It has nothing to do with exercise today. That's almost nothing compared to the calories out. We're looking at heart function, lung function, organ function, what your brain is doing, thermal regulation. Ever notice when you go on a diet or you're fasting or you're just shy on calories for a while, you're like cold all the time? Get a little bit heavier, you're like sweating all the time? You can't just put your mind to it, like, ooh, let me get super cold. Oh, yeah, that feels terrible. You're burning a ton of calories when you're shivering. You're burning a ton of calories when you're sweating, not by sweating, but you're sweating because of that. Now, this one is particularly important, maintaining your ion gradients, maintaining your ion gradients. You can't do that by way of will. That's not something you can do. And it changes depending on hormones, right? So we've all met somebody or struggled with this ourselves, or at least know someone who knows someone who has hypothyroidism or hyperthyroidism. Hyperthyroidism, I mean, like, man, you can't stop sweating when you're just like trying to shed as many calories as possible all the time. Very, very thin usually. Hypothyroidism, you're wearing like 9 blankets, right? It's not calories in, calories out. What we're looking at is, you know, the thyroid hormones. So, you know, again, you can't just be like, all right, let me get some thyroid. Got it. You can't do that. You are in no control of your calories out. You can control your calories in, but when you look at how many calories you're spending throughout the course of the day, basal metabolic rate, that's it, right? That's the bulk of all your calories, and you can't control that. Your body makes huge sweeping changes based on calories in. Okay, now here's neat non-exercise activity thermogenesis. This is just like, are you twitching? Kevin's like pumping his leg right now. That's good. Don't stop, right? Just because I'm pointing it out, keep it up. That's super helpful. Neat, you're burning way more calories than you are with exercise. You know, the people who are just like twitching and flicking around a pencil and pumping their leg and just all of that stuff, that's more important than exercise. When you look up the studies and do these comparative analyses of when people gain weight, when people lose weight, NEAT is more important than exercise. And then thermic effective food, that's bigger than exercise. So here's what people are doing when they're trying to control their body weight, calories out, right? It's so close to nothing that you can round down to exactly nothing without sounding foolish. This, the huge one, even meat is really controllable by like thyroid hormones and things like that. But testosterone and epinephrine and thyroid hormones, all of these things change in response to calories in. Calories in, calories out. There's like no creature, I can't say that, I'm not like a biologist, and like I'm not a zoologist. Certainly for humans, that recipe has never been applicable to an upright bipedal creature since like Lucy, Australopithecus, like the ancestors of that. Now, just look at the Biggest Loser. This, and we all know what this is, right? A 1 million pound person goes on and loses like almost 1 million pounds in like a day. How ludicrous this show is. But notice that you've never seen a reunion show in which the people who lost all that weight come back and said, look at my huge jeans. They're still huge on me. Notice that's never happened, right? So they did a follow-up study. 16 Biggest Loser competitors originally investigated. 14 of them showed up for this follow-up study. Now, normally, I don't know what this means, but normally when people don't show up, what that means is they're embarrassed. They don't want people to know. It's like, I don't want people to know that I gained, 2 million pounds back. I'm not saying that's what happened here, but generally, that's the case. I don't know what the case is here. But you look at stuff like physical activity. Okay, baseline was very low. Okay, and then end of the competition, baseline physical activity, high. It's the same as it was. This is 6 years later. Same amount of physical activity. What happens to the basal metabolic rate? Resting metabolic rate. It was high at baseline, 2,600 calories. And then it was 2,000 calories. Now it's 1,900 calories. It's even lower. Remember, basal metabolic rate, they're down like 700 calories. They're down 700 calories a day in their basal metabolic rate. Obviously, they gain like just a ton of weight back. Everyone does. Calories in, calories out. Their diet is more controlled, right? They're exercising more than they ever have, and they're gaining weight. Calories in, calories out. That's ludicrous. Yeah.
Just their weight change.
Yeah, so if you look at their weight change, so they went from like percent body fat, they went from 49.3 down to 28, and then back up to 45. But again, look at their their physical activity is great. Their metabolism is what crashed. And so what you see, their age obviously goes up 6 years, because it's been 6 years, but their weight and BMI and body fat percent, all of these things, it's great. They do a crash. And what crashes with them? Their metabolism. There are ways around this, and that's what we'll talk about on Wednesday. How do we avoid doing this? Because very few people succeed in weight loss. It's easy. It's super easy to lose weight really fast. If it's like, I have a month to lose a ton of weight, okay, well, you'll be heavier in 2 months than you are today probably, but we can get you there. But what you want to do is pick an ideal weight for you and be there forever. And there are ways to do that. There are ways to accelerate fat loss, and there are ways to preserve fat loss. Preservation is where most people fail because they go about it wrong in terms of the actual weight loss itself. So, ATP hydrolysis, we got to put it back together. There's a bunch of ways that we put it back together. Oxidative metabolism, we're going to go start to finish today, from lipolysis to the electron transport chain. How fat is consumed. Now, carbs are sort of ultimately consumed, can be there as well. And we can also talk about ketones as a source of fuel. But the adenylyl kinase reaction, just remember ADP plus ADP, you get an ATP and an AMP. Adenylyl kinase is the enzyme. Creatine kinase reaction, creatine kinase enzyme. We take phosphocreatine or creatine phosphate and an ADP and a hydrogen ion, and we donate that phosphate to the ADP to make it ATP, and then we plug up the creatine with the hydrogen. So the creatine kinase reaction, the adenylyl kinase reaction, these are very fast ways to reconstitute our ATP. Glycolysis, and toward the end of the semester at some point, we're going to go through glycolysis more step by step. And when we're talking about performance and fatigue and the reason we stop exercising. But most of these diagrams, hexokinase is what they list first. And we're human beings. Hexokinase isn't always what we eat. We're not yeast, right? So we don't always use hexokinase. We do if it's glucose. But if it's glycogen, we use a couple different enzymes to get the G6P. Glycogen phosphorylase and phosphoglucomutase. We have a different road into glycolysis. At the end of glycolysis, right, we get a couple of pyruvates. And you can do what anaerobic, right, where you just convert to lactate, or you can pump it into the Krebs cycle, tricarboxylic acid cycle, citric acid cycle, or whatever you want to call it. It's all the same thing. And so we'll talk a little bit about that today. But it's just, this is a quick little summary of what it is. Acetyl-CoA, this little 2-carbon thing. Okay, it binds to this 4-carbon thing, oxaloacetate. It becomes citrate, a 6-carbon thing. That undergoes a series of enzymatic changes and reemerges as a 4-carbon thing, oxaloacetate again. So the Krebs cycle, because a 4-carbon thing does a bunch of stuff and returns to life as itself. Binds to acetyl-CoA, becomes citrate, undergoes a bunch of changes, becomes oxaloacetate again, ready to accept another acetyl-CoA. What happens in the presence of a low-carb diet? Wait, what? Ketosis. So you go into ketosis, and what here? Let's say we're in the liver and we're doing a Krebs cycle in the liver. What happens to create ketosis, to create a ketogenic diet, to generate ketones? A ketone is formed, or forming ketones, out of two carbons. We take these acetyl-CoAs and bind them to each other to make our ketones, because we strip oxaloacetate out for gluconeogenesis in the liver. We take that to make carbs out of non-carb sources, gluconeogenesis. Let's take oxaloacetate out, and then the acetyl-CoAs show up looking to bind to oxaloacetate. There's nothing to bind to, so they bind to each other, and we're going to get some ketones that way. But ordinarily, this is how it works. Now, this 2-carbon thing, there's one of the carbons, there's the other carbon. You're releasing this carbon dioxide. Along the way, and we'll talk about this, you create an NADH and an FADH. You create those along the way. Now, you can make acetyl-CoA from multiple things. Acetyl-CoA has multiple fates. You can make it out of your pyruvate at the end of glycolysis. You can make acetyl-CoA out of that. Most of your acetyl-CoA, you're making out of fatty acids. Acetyl-CoA has a lot of fates, but most of it is the Krebs cycle. We'll focus on those most. Okay, so here's the 2-nippled man. Let's say third, because maybe there's one on the other side. Lipolysis, we're lysing our fats. Lipolysis, lipolysis, lipid lysis, fat lysis, we're lysing our triglycerides. A triglyceride, or triacyl glycerol, is that 3-carbon glycerol molecule with 3 fatty acids connected to it. So here's your glycerol, fatty acid, fatty acid, fatty acid. You have a big chemical compound. And what lipolysis is that. You take a triglyceride, you do one at a time. You don't just take the hash and do all 3 of them at the same time. But you lyse them, you release them. You get 3 fatty acids. It was all bound up, right, as a triacyl glycerol. And then you split it up into these 4 component parts, the glycerol, and 3 fatty acids. So this is, again, how it works. Now, you can't, if you're gonna burn fat, literally burn fat, think about burning wood in a fireplace. You don't take a huge hunk of trunk and put that in the fireplace. You gotta split the wood first. This is too big, you can't burn that. It'll never catch, it even fits in there, right? So you gotta chop it up into 4 pieces. Get 3 fatty acids out of it. And those little pieces Those little components, that's what you burn in the fireplace. Fatty acids, those are what you burn in the mitochondria. So epi or norepi, but this is a cell right here. This is a adipocyte. This is a fat cell. Everything inside of this is inside of the cell. This is the membrane right here. This is the outer layer of a fat cell. In here, we see a triacylglycerol or a triglyceride. A diacylglycerol, meaning we've separated one of those fatty acids, and there's two of them left up there. There's multiple fates for this. You could use diacylglycerol kinase and convert it to phosphatidic acid. We'll talk about that soon with mTOR. That's a very anabolic thing to do. You can turn on mTOR with phosphatidic acid by converting a diacylglycerol to that with diacylglycerol kinase. But lipolysis, hormone-sensitive lipase, is going to lyse a fatty acid from it, and we get monoacylglycerol. So there's the glycerol in one fat, and then monoacylglycerol lipase. Let's get rid of that last fat, that last fatty acid. So epinephrine, this is systemic. Epinephrine from the adrenal glands. That is a circulating endocrine hormone, an amine hormone made from one amino acid, but it is a hormone in circulation. Norepinephrine or noradrenaline. Epinephrine and adrenaline, same thing. Norepinephrine and noradrenaline, same thing. So the nor, this is coming from your nerves, predominantly, almost all of it. It's coming from your nerves. Fat is innervated. Nerves connect to your fat. So whatever, your stomach and your whatever, it is innervated. There are nerves connecting to it. And so you can get a systemic release of epinephrine, just send it there through the blood, or norepinephrine, send it there through the nerves, and either of those binds to a beta adrenergic receptor, and you will get activation of adenylate cyclase or adenylol cyclase. You'll see both spellings. Adenylate cyclase converts ATP into cyclic AMP. Remember, cyclic AMP binds to pKa. pKa gets activated that way. And pKa phosphorylates a ton of things. Protein kinase A. A kinase attaches phosphates to things. And so one thing it's attaching phosphate to is hormone-sensitive lipase. Now, really hormone-sensitive lipase is doing this one. It's working on the diacylglycerol. Not as much activity here. Much more activity here for hormone-sensitive lipase on the diacylglycerol. Adipose triglyceride lipase is much more responsible for this. But so one of the things that pKa activates is that hormone-sensitive lipase. And this is adenylyl cyclase, AC, adenylyl cyclase. It takes your ATP, 3 phosphates, converts it to cyclic AMP, cyclic AMP. So there's regular AMP, which does not activate pKa. This is not an activator of pKa. Cyclic AMP is what activates pKa. And you can convert cyclic AMP into regular AMP with phosphodiesterase, PDE, phosphodiesterase. What activates phosphodiesterase? PKB. Alex, are you 2, 3? Oh, 19. PKB, so PI3K, PKB, we're activating PDE, that's insulin signaling. We're activating phosphodiesterase PDE. PDE converts cyclic AMP into regular AMP. Regular AMP does not activate pKa, so we do not get phosphorylation of hormone-sensitive lipase. And we do not get activation of pyrrolipin. What's pyrrolipin? Yeah, are you guys on the same team?
Yeah, I was doing it.
Okay, you were both, yeah, there was a mime and a speech. You're both on team 3? Yeah. Okay. 0.016. So the coat around the lipid droplet, right? The coat around that. So pKa is going to phosphorylate that too. So we have beta adrenergic receptor, epi or norepi. Let's bind to that thing. And we are going to activate adenylol cyclase, it says here, adenylyl cyclase, who cares? Convert ATP into cyclic AMP. And cyclic AMP would be activating PKA, but phosphodiesterase comes in and breaks it down. The whole thing is stopped, and so nothing downstream happens. If you put a roadblock up somewhere, nothing downstream follows. So this would be that lipid droplet with the coat. Here's perilipin. You have to phosphorylate the perilipin for hormone-sensitive lipase to gain access. So the triacylglycerol, or a triglyceride, a ton of these triglycerides live inside of this lipid droplet. But for the hormone-sensitive lipase to gain access, you have to phosphorylate the coat. You open the coat, and the enzymes can get in and do lipolysis. But when you PKA, so it does increase adipose triglyceride lipase, that one that does the first, the triacylglycerol. Let's convert that into a diacylglycerol. Let's lyse the first fatty acid. Adipose triglyceride lipase. PKA does seem to increase it, twofold, but hormone-sensitive lipase, 26-fold. PKA signaling really increases hormone-sensitive lipase. Yeah, it increases adipose triglyceride lipase, regulated a little bit different. Long-term effect of insulin. So short-term effect of insulin, get insulin in your system, and you can't do lipolysis because you can't activate PKA, which means you can't phosphorylate hormone-sensitive lipase or perilipin. In the presence of insulin, you cannot phosphorylate perilipin or hormone-sensitive lipase. Long-term, insulin is decreasing the expression of adipose triglyceride lipase. You get less of it. The first enzyme that initiates lipolysis in the presence of insulin, in the presence of more and more and more and more insulin, that's what this is. Increase the insulin, more, you're getting less adipose triglyceride lipase. So that's a little bit longer term. That's genetic. That's a genetic expression. The behavior of your genes. It's not like it's genetic, like it's assigned to you at birth. The behavior of your genes. You are creating less of that enzyme. So this should make sense to you. Beta adrenergic, adenylyl cyclase, right? Adenylyl cyclase converts ATP into that, cyclic AMP. Phosphodiesterase 3B, there's a bunch of kinds of phosphodiesterase 3B, that's the fat one, is going to block cyclic AMP by converting into regular AMP. There's hormone-sensitive lipase, there's perilipin, PKA phosphorylating both. And this is what we're going to talk about after we do the kind of strategies of fat loss. We'll talk about mTOR signaling. From insulin, there's insulin receptor substrate. This is our receptor, PI3K. That converts PIP2 to PIP3. Phosphatidylinositol bisphosphate, or who cares, 2 phosphates. Phosphatidylinositol dose phosphate is fine. Whatever, 2 phosphates into 3 phosphates. Phosphatidylinositol triphosphate. And that allows PDK to activate PKB. PKB phosphorylates a lot of stuff. Just like PKA phosphorylates tons of things, and those things are catabolic. Those things associate with mobilization of energy substrates. PKA gets energy substrates on the march. Let's burn stuff, that's PKA. PKB is let's get big and store stuff.
How much cyclic AMP does it need to explore, right? 4. Okay.
Yeah, so 4 of these guys to activate PKA. You'll be making a ton of these things, and so just kind of the flood coming in. Now, as we see more and more insulin, here's your fasting insulin as it gets higher and higher and higher and higher and higher. You see the non-esterified fatty acids, they're like free fatty acids, getting lower and lower and lower and lower. You are not mobilizing fatty acids in the presence of more and more and more and more insulin. You're having a harder and harder and harder time mobilizing fat. It stays put. Long-term insulin sensitivity problems, things get a little bit different. Relatively short-term and medium-term, this is what it looks like. Where the more insulin you have, the harder it is to release fatty acids. Insulin is the single biggest known opponent to fat metabolism. Endogenous opponent. Of all stuff that your body is just making, insulin is the biggest opponent of it. Now, you mobilize all these fatty acids through lipolysis. What do those fatty acids look like? What's the fate of those fatty acids? They're different lengths. There's short chain fatty acids, medium chain, long chain, very long chain, ultra long chain. The way people categorize these things, if you just think like short chains and medium chains, 1 to 12 carbons long, carbon, 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 carbon. Remember, there's just these long strings of carbon. If you're up to 9 carbon atoms, let's get under 10, you're going to be liquid at room temperature. So you can have a saturated fat. We think of saturated fats as being solid at room temperature. But if it's a really short chain fatty acid, that's going to be but carbon numbers from 1 to 6, generally short chain fatty acids, we call them short chain, 1 to 6 carbons. Medium chain, again, people will categorize these things a little bit differently depending on the publication. But the 7 to 12 carbons, medium chain fatty acids. Long chain fatty acids, that's what you see in the bottom most. You see short and medium too, but there are 16 and 18 carbons. Those are the bulk of all fatty acids in your body that you're metabolizing. You go through lipolysis and beta oxidation, 16 carbons long and 18 carbons long. Those are the real fatty acids that make up your basal metabolism. The bulk of your basal metabolism is 16 carbon and 18 carbon fatty acids. So just 3 of those make up about 85% of all triacylglycerol. Just a few of those, and there are more 16 and 18 carbon ones. Again, you can get into these very long chain ones, the very long chain fatty acids, and then you get the ultra long chain fatty acids and whatever. We're not really doing mitochondrial consumption of these. The peroxisome is where we're dealing with some of these. So what we think of for metabolism, for your basal metabolism, is long chain fatty acids, mostly 16 carbons and 18 carbons long. And to do that, beta oxidation, we're going to take that 16 carbon, let's say, fatty acid, and we're going to beta oxidize it, which means cut a 2 carbon acetyl-CoA from it. You chop off a little 2 carbon chunk, that's an acetyl-CoA, and then you still have your acyl-CoA, your fatty acid. You still have your acyl-CoA. Now it's 14 carbons long. You cut off 2 more carbons, and now it's 12 carbons long. Cut off 2 more, and now it's 10. So if you have a 20 carbon fatty acid, how many acetyl-CoAs do you get from it? 10. You get 10. If you have, what was that, 20? If you have an 18 carbon fatty acid, the acyl-CoA, how many acetyl-CoAs do you get? 9. If you have a 16 carbon, how many do you get? 8. If you have a 17 carbon fatty acid, how many do you get? It's actually lower. The last one, you get 5. I'll show you this. So instead of doing this with the last one, you actually get the same. Right? You do this. This is an entry product into the Krebs cycle elsewhere, and then that's your acetyl-CoA. If you have an odd carbon fatty acid, they're rare. You're not doing this much. Those are rare. But you don't do that. You don't cut it into 2 and like have 1 carbon go float off and it's carbon dioxide. That doesn't happen. You cut it into a 3 carbon compound. And that's a different entry product into the Krebs cycle. I'm not doing the math. I think so. I think it's 7.
It's just 7, but one has 3 carbon.
Maybe it's 6. I don't remember how many we said to begin with. What was our? If you have like 16. Okay, let's draw the 14. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14. So that's what we have. So if you have a 14 carbon fatty acid, you get 1, 2, 3, 4, 5, 6, 7. Right? If you have a 15, 1, 2, 3, 4, 5, 6.
Oh,
I'm just getting.
oh,
Because that does not become an acetyl-CoA. So add an extra carbon, you get 1 fewer. I didn't feel like doing the math as I was just standing there. But drawing it on the board is much easier to think through. So there's a lot. You don't memorize any of these, don't even write it down. But there are a lot of enzymes, proteins that facilitate chemical reactions, proteins that facilitate this process of beta oxidation. There's a lot of them involved. This is what it looks like. Okay, you have your fatty acid. It's outside of the cell, right? So you're exercising, and the fatty acid, let's say you're doing cardio on with your legs on a treadmill. The form that is not like this. You are metabolizing fat from all over your body. It's not like, well, I'm just hitting fat, intermuscular fat in the quads and glutes and hammies. Like, that's not how it works. Fat's coming from everywhere and being sent to the furnace. That's what lipolysis is you release fat from elsewhere and deliver it to the metabolically active tissues. So you get a fatty acid, it has to enter that cell, the metabolically active cell. The fatty acid has to enter it, and you get it in there, convert it into an acyl-CoA by attaching a coenzyme A. Now we have an acyl-CoA. We'll do step by step in just a second. I'll go a little slower. Now, to get it inside of a mitochondrion, this guy, to get it inside of one of these, you need to attach carnitine to it. Attach carnitine to it. So we attach carnitine to it. We get acyl carnitine. Acyl carnitine. This is just your free fatty acid with carnitine on it. And coenzyme A. We got to get it into the mitochondria. So we shuttle that in. At the same time, we shuttle free carnitine out. Just think of it as like a sodium potassium pump. We're exchanging. Free carnitine is coming in. Acyl carnitine is coming in. Acyl.
carnitine is a transporter.
Yeah.
What's the transporter?
Well, the it's a CACT is the actual transporter. I'll show it to you in just a second in more detail. And so carnitine, think of it as like the usher, like I don't know. I have bigger text coming up. You don't need to split. This is just for this. This is the article. That's the caption for it. But acyl carnitine, we convert back into acyl-CoA, into that fatty acid. Here is actual beta oxidation. It occurs in the mitochondria. At the end of it, we get our acetyl-CoA. Right now, the acetyl-CoA, that can enter the Krebs cycle or ketogenesis. Bind them together. Let's make some ketones. So let's go step by step. Same authors, is actually just earlier, but it's a less pretty diagram, but I actually like this one better. So fatty acid transport proteins. Let's get it into the cell. Let's get the fatty acid into the cell, and at that same time, we're going to bind a coenzyme A to it and get our acyl-CoA. So an acyl-CoA, just think of this as your fatty acid, your acyl-CoA. The next step is to make acyl carnitine, so the CPT1, to make your acyl carnitine. Once you have that, you're actually able to get it in, but that CACT, carnitine, acyl carnitine, translocase, that's what does the exchange. CACT does the exchange, the acyl carnitine comes in, and the free carnitine comes back out. Now we have our acyl carnitine, but acyl carnitine is not what we oxidize. We don't oxidize acyl carnitine. We oxidize the acyl-CoA. So CPT2, we're going to remove it. We're going to free up that carnitine. So now we have free carnitine and an acyl-CoA. That permits CACT to shuttle that carnitine out and a new acyl carnitine in. So we keep using carnitine over and over. Carnitine comes in, escorting an acyl-CoA in, and then carnitine goes back out to escort a new acyl-CoA. So carnitine just works, right? It has like, I don't know what to give an analogy. It's like it has, it's a footman, yeah. If it's like Down Abbey or something. Or like if you're somewhere, there's like a doorman who says like, no, you can't come in. And somebody like inside the club is like, no, they're with me. And they get past the doorman. In that analogy, CACT, doorman. Carnitine, the favorable person inside the club who like comes out, no, they're with me. They can come into the club. That's carnitine. And then they see someone else to be in the club. No, they're with me too. They're with me too.
Didn't you just say that person used to be like Usher?
I have a picture of Usher. Yeah. So that's really how it works. Is carnitine. Or think of like at a wedding, somebody escorting you to your seat, right? Like, Great Granny Roth shows up and she's like, where am I sitting? And like the young usher is like, no, here, let me take you to your seat, right? Take you to your seat. Carnitine doesn't just like sit there a whole wedding, staring at Granny being creepy, right? Carnitine's like, no, there's someone else I need to usher. And so carnitine leaves the pavilion or wherever a wedding is and ushers in a new acyl-CoA. Now you sit here, you're going to be chopped up. You're here for the slaughter.
Can there be like too much fatty acids that weren't and then carnitine like overwork?
Yeah. It's a good question. I'm going to have to stare at you like a bad usher while I think. Yeah, can I come back to that later? So, like with all things, we'll talk about if you have too much end product, if you have too much acetyl-CoA, what's going to happen to fat metabolism? We'll talk about some of those things, but if there's too much something, there's almost always a situation in which it's saturated, in which there's a saturation, but I'm unfamiliar. I would have to look it up. I'm unfamiliar with the context in which you're going to see there being too much. It could be something. This is just speculating, but there could be something like when you look at late stage type 2 diabetes, right? Type 3 diabetes. If the insulin receptors are insensitive, if the insulin receptors are insensitive in a fat cell, you're really not blocking lipolysis. You actually do get a lot of release of those lipids. And so maybe in a context like that, we're going to see this increase, really greasy blood. I don't imagine short-term fasting would associate with anything like that. In short-term fasting, you're going to get a ton of this beta oxidation, but you're just going to create a bunch of ketones. You're going to increase the amount of ketone generation. Okay, so we were at carnitine. Is that where we were?
Where's carnitine made? In mitochondria or?
From amino acids. It's a drug.
If you take carnitine as a supplement for fat loss, do you just have more ushers?
You have more ushers. Now, originally, it was, and we'll talk about this on Wednesday. There's studies on this. Originally, it was just like, well, it's calories in it, and eat anything with calories, you can see your metabolism increasing. And then it was like, whoa, we have pathological conditions, HIV-related muscle wasting or something like that, where you're going to lipodystrophy, things like that. And you're going to start to see a depletion of carnitine, and it works great there. But it turns out that it actually does have effects in lay folks too. So there's some evidence for it. We'll talk about that on Wednesday.
Isn't it just a protein though? You take it and it just goes body digestion?
There are fates to amino acids, and carnitine is one of the fates that this role here. But it depends what your metabolism calls for.
So where do we inject it?
What do you inject
Yeah.
it?
Yeah.
This is getting more immediately applicable than I was expecting. So we'll talk about the literature about carnitine is one of the things that has some evidence for effectiveness. So I have no idea what's over here, but so I think we're to the actual beta oxidation step, where we get the acyl-CoA and then we oxidize it, right? We do that thing where we chop off little 2-carbon chunks. Just saying like what we're going to get out of it over here, what it yields. But here are the steps. So step 1, and there's JP's usher. Step 1, convert the fatty acid to an acyl-CoA, right? Attach coenzyme.
That is beta oxidation is my.
Yeah. So this is just when we get to beta oxidation on the next slide. So this is pre-beta oxidation. We have this 4 steps of like we've just gone through lipolysis and delivered this 14 carbon, 16 carbon, 18 carbons, probably a 16 or an 18 carbon fatty acid to the cell. How do we get it in there, right? Beta oxidation takes place inside of a mitochondria. That's where beta oxidation takes place, inside of mitochondria. Well, how do we get it inside of there so we can oxidize it? Attach an acyl-CoA, right? Step 1. Don't write down enzymes. Attach coenzyme A. Convert that acyl-CoA to acyl-carnitine, meaning attach carnitine to it. Attach Mr. Usher to it. Mr. Usher can cross the membrane of the mitochondrium, go into the from the cytosol into the mitochondria. You're going to move the free carnitine out at the same time the acyl carnitine moves in, and then you remove that carnitine, so that's free to go back out. Usher can go usher someone else to the seat, and then you beta oxidize. And so this is what beta oxidation looks like. The first step is a dehydrogenation, dehydrogenase, a dehydrogenase. Remove hydrogen and oxidation. There's 2 steps that are dehydrogenation. It's dehydrogenation, and then hydration, and then dehydrogenation, and then you lyse it. So the first step, notice the H2s here. Notice H without the 2, right? FADH. That's a carrier compound. That's for the electron transport chain. So we dehydrogenate it at the first step. Second step, just add some water, right? Hydrate it. Hydration, second step, just add some water there. The HO, the H, right? H2O. So step 2, add water. Step 1, dehydrogenation. Step 2, add water. Step 3, dehydrogenation. Step 4, you lyse it. Just like ATP hydrolysis, you use hydro, you use water to lyse. Thiolysis, right? That's what you're using to lyse it. That's just a sulfur, a hydroxyl group is an OH, like an alcohol. Cholesterol is a steroid alcohol. There's an OH. That OH, if you take the O and replace it with sulfur, that's what you're using to lyse this acyl-CoA. So you lyse off an acetyl-CoA. Sometimes it's acetyl-CoA, especially if I'm here in Texas. So your acyl-CoA, if that's like 16 carbons, now it's 14 because you just lysed an acetyl-CoA, a 2-carbon compound from it. Eventually you get down to nothing, right? You just lyse it until there's nothing remaining. And again, rare, but there are odd carbon acyl-CoAs. And if you get one of those like 17s, 15s, if you get an odd carbon acyl-CoA, you are going to convert it. Propionyl CoA, you're going to end up making succinyl CoA. So it's just this Krebs cycle intermediate.
Where's the if you're depleting your glucose and you're focusing on fat as a method of energy, where do you get the acetyl-CoA Gluconeogenesis or?
Yeah, you'll have a ton of gluconeogenesis. So you're making acetyl-CoA from, I mean, most acetyl-CoA you're just getting from fat. I mean, so that's like your fat metabolism is really enhanced, but you're going to get like oxaloacetate, right? That's gluconeogenesis. But in a ketogenic diet, fat metabolism, people will say things like, what is, what's a ketone? Like the incomplete breakdown of fat. That's just the expression everyone says, the incomplete breakdown of fat. Well, you break it down completely, you just, you don't send it into the Krebs cycle. Because you might strip out some oxaloacetate in the Krebs cycle. So you're going to break it down into all the acetyl-CoAs. You'll do it way more. So fat metabolism, the rate of lipolysis, if you look up a ketogenic diet compared to a normal diet, the rate of lipolysis is outrageous in a ketogenic diet. You actually are capable of mobilizing fat way faster. You can burn way more fat on a ketogenic diet. And you get a ton of acetyl-CoAs, but you don't really run them all through the Krebs cycle because you strip out the oxaloacetate. And so then you start binding acetyl-CoAs to themselves to create ketones.
And then the oxaloacetate comes from pyruvate.
Yeah, you can make oxaloacetate out of other stuff too. But I mean, normally you just out of knowledge, right? You just create it out of itself. Okay. But there's a lot of here. I think that's like the next slide where I show in the Krebs cycle. Now there's a ton these can leave the Krebs cycle. Stuff can enter the Krebs cycle, just like succinyl CoA can come out of fatty acid. The Krebs cycle has a lot of inputs and outputs, inroads and outroads, and stuff like thermogenesis and immune function, stem cells. There's a lot of metabolic regulation that occurs in the Krebs cycle. So it's not just, let me take an acetyl-CoA and chop it up into carbon dioxide. It's not just that. There's a little bit more to it. But the connection with the Krebs cycle and oxidative phosphorylation, OXPHOS, oxidative phosphorylation, which includes ATP synthase, the very end step where you're creating much ATP. The electron transport chain is just these four little hubs right here. The electron transport chain, oxidative phosphorylation, includes ATP synthase. We're creating in the Krebs cycle NADH, FADH, and those are feeders. Those are sort of middlemen, sort of cutoff players for the electron transport chain, which is where this ends, where this metabolism ends. So you have your acetyl-CoA, a little 2-carbon compound, and it binds to the 4-carbon oxaloacetate to make that 6-carbon citrate. And really, again, NADH, NADH, FADH, NADH. This is what's coming out of it that feeds into the next step, that feeds into the electron transport chain. So just the reducing agents means donating electrons, right? So these things, do you know Wallace and Gromit? Okay, so Wallace is the one who talks, he's like the star of the show, his name comes first. I'm Wallace. That's NADH, that's the bigger player. That's the headline. It's the more important character. Gromit is the cute one, the lovable one, is everyone's favorite character, but Wallace is the headliner. NADH, it's more important. NADH is more important. You create 3 of those. But you're just donating electrons. So Gromit, that's your FADH. Again, you're donating electrons. It's a reducing agent. Reduction is you're gaining an electron, is a reduction. If you're a reducing agent, you're the one donating that electron. So FADH, NADH. NADH is more important. Now, if you listen, these are my fingerprints from the last time I taught this exact thing, which is probably like to your class. I haven't taught it in years. If you listen carefully, like to the walls, you'll probably hear You probably hear like some mice scratching around in there. You know, mice are there in the walls. Mitochondria have that space, you know, that mouse that's in the bread bag. But mice live in the walls. Mitochondria have this inner membrane where stuff can live. This inner membrane where stuff can live. And the purpose of the electron transport chain is not just that electrons like go down, you know, this chain, right? They're attracted to, progressively attracted to the next step. It's that you pump hydrogen ions into that space between the walls. And so complex 1, and I'll show you a little diagram of this, but complex 1, this is where NADH delivers its electrons. NADH, Wallace, delivers his electrons here. At step 1, you pump, let me put up a new like sweaty hand mark. You pump hydrogen ions into the inner membrane space, into that space between the walls, not outside, but between the inner wall and the outer wall, that little narrow space between the walls. You pump 4 hydrogen ions, 4 protons. Step 2, no protons, hydrogen ions, no protons, go into that inner membrane space, but this is where FADH delivers its electrons. So notice that NADH comes first, and you get 4 extra protons pumped into that space between the walls with NADH. NADH, you get a bigger proton concentration in the walls. Step 3, another 4 hydrogen ions, protons, hydrogen ions are pumped into that space. Another 4 at complex 4. And then we're going to bind it to oxygen, right? All these hydrogen ions and the electrons are going to bind to oxygen at the end. So you make water at the end of the electron transport chain. You donate these electrons early on, and they go down this little chain, and you're pumping hydrogen ions. That's the important part. You're pumping hydrogen ions into that space between the walls, and you create a huge gradient. A huge gradient. So this is that inter-membrane space over here. And the space between membranes, and you're pumping all these hydrogen ions at step 1, step 3, and step 4. And step 1 is where NADH delivers. Step 2 is where FADH delivers the electrons. Then you go down a little bigger. You bind the electrons and hydrogen ions with oxygen to create H2O, to create water at the very end. But the main thing you do is create this huge gradient of hydrogen ions. And that powers ATP synthase. And that's how you get all your ATP back. It's like a big mill. Now, if you have, say like hyperthyroidism, something like that, you have some condition in which your basal metabolism is just roaring, you can be leaking these things and not creating ATP out of them. And so you are really going to be burning more calories as part of your basal metabolism. If this isn't working very efficiently, if you're leaking them, your basal metabolism is very different. And that's something, again, you're in no control over how well this system functions.
Where would it leak through?
I don't know the exact site of leakage of where they're leaking through. But the way it pumps those, just think of like a mill, like a wind turbine or a water mill. And that's how ATP synthase is powered to reassemble our ATP. So this is your ATP synthase, right? ATP is coming here, or the hydrogen ions are coming through, and that's powering it to create your ATP. Now, in orange is the new black. I watched seasons 1 and 2 when it was new, so that was a long time ago. The last time I saw this was probably like this episode, or however many seasons there are, however many episodes there are in the season. But this is Norma. Does anybody know who this character's name is?
I also watched only the first one.
Yeah, the first 2 seasons, I'm like, okay, I got it. Whatever. I read the book and that was fine. And then it was like nothing about the book anymore. Whatever, this character. So So that's not V, but V is this lady that she's trying to kill, and she's collecting apple seeds to poison a character named V. And this is what this character says, word for word. I like heard it at the time, and I like rewound it and watched it again and wrote it down. You know how many seeds you're going to need to make a lethal dose of arsenic? More than all the apples ever given to any teacher that you've ever known. Okay, that's the line. That's the exact line, word for word, on the show. Now, cyanide is an inhibitor of cytochrome C oxidase. Okay, so that's the fourth complex in the electron transport chain. So that transfer electrons from cytochrome C to oxygen. So at the very end of the electron transport chain, the transfer of those electrons to the oxygen to make the water. Cyanide is an inhibitor, and apple seeds don't have any arsenic at all, but you can get cyanide out of them. They have amygdala. Metabolically, that releases cyanide. So you can get cyanide out of apple seeds, but it really, I mean, it takes like contaminated drinking water to get arsenic. So that's not what you're gonna get. But maybe, I don't know, like 20 apples worth of seeds or something like that. I think teachers will probably receive more than that. But pyruvate, remember the end of glycolysis, we get pyruvate. How do we convert pyruvate into acetyl-CoA? If we're not going to convert pyruvate into lactate, we're going to convert it into acetyl-CoA, entry product of glycolysis. That's what arsenic is inhibiting, the pyruvate dehydrogenase complex. If you have contaminated drinking water, what you're obstructing is a conversion of pyruvate into acetyl-CoA. So you get a ton of lactate. Okay, that's everything for fat metabolism. That's going from like, our tum-tum to water. That's like, we have taken fat and now we have water at the end. What questions do we have from step 1 to step whatever it was, end? It appears to have recorded. Yeah, so on Wednesday, if you have questions, we'll cover those things, but we'll talk about techniques and strategies and interventions and supplements and everything.
Can we go back to where acetyl-CoA comes into play with fat oxidation?
Maybe if you go back to this slide.
Yeah. So it's just going through its own version of the process.
So you have acyl-CoA. You make it as this as you move it into the cell. Right, this is your out of the cell. This is like you're in the blood and you're migrating and you get to the cell. You bring it. You have this protein that facilitates the transport across the cell. And at the same time, you attach a coenzyme A to it, and so you get your acyl-CoA. So that's essentially the fatty acid. And then you do the carnitine stuff. And acyl-CoA can't just go in. Just like a fatty acid can't just wander in. An acyl-CoA can't just wander in. So you need this transport protein. With acyl-CoA, you attach the carnitine, and then you can bring it in. Just for the electron transport chain, the 4 complexes. Cytochrome C oxidase is the only complex to remember. That's NADH delivers a complex 1. FADH2 delivers a complex 2. Complexes 1, 3, and 4 pump protons into that space. And at the end of that, that's the electron transport chain. The electron transport chain does not include ATP synthase. That's oxidative phosphorylation, includes everything. But the electron transport chain is just those 4 complexes. ATP synthase, yeah, that's included in oxidative phosphorylation, but that's the step after the electron transport chain. Electron transport chain is complex 1, 2, 3, and 4. Complex 4 is cytochrome C oxidase. That's the thing cyanide inhibits.
So you're making acetyl-CoA from acetyl-CoA,
Yes.
and then you do.
And then decrypts. Yeah, so you take an acyl-CoA, which, you know, again, it's probably 16 or 18 carbons. And that acyl-CoA, just you chop it up, if it's 16, you make 8. Right? You make 8 of these acetyl-CoAs. And those are the feeders into the Krebs cycle. And yeah, in a ketogenic diet, you just tons of this.
What's FAO?
Fatty acid oxidation. Yeah, so fatty acid oxidation, just like everything.
Yeah. So what do we inject from?
We'll do we'll do all the supplements. There are quite a few supplements that work. There's quite a few, and we'll talk about all of them on Wednesday. Every little trick that we can do. All right, so I will see everyone